Welcome. We're going to go ahead and get started. I'm Robert Urban. I'm the Executive Director here at the Koch Institute. We're delighted to have all of you here with us for another event with our Within Sight lecture series. This is a very special night for us for a number of reasons. I'll go through those with you as we get started. We have a number of special guests in the audience. We have members of our Leadership Council, a very important group for us to help us bring the Koch Institute to life in the first instance, to keep it on track on a daily basis. We have special guests from the Silverman family who have been, throughout this launch of the Koch Institute, truly remarkably helpful in bringing the voice of the Koch Institute out to the community. We're very grateful for them. They're here visiting with us today. We also have members of the Within Society, Benefactor Group. This group helps us raise money and create the gallery programming that we bring to you. We hope you'll consider looking at the material that you've been presented tonight and consider joining that group if you're interested. It is a mechanism by which you can gain access to some of these prints, for example. We're also durably grateful to Charles B. and Ann L. Johnson for helping us create the gallery in the very first instance. It was through their philanthropy that we were able to build out the gallery and put it on the front of the Koch Institute. The Within Sight lecture series is an offering that we've created to help us bring the Koch Institute out to the community, to help bring an approachable, hopefully informative, and I'll also really hope inspiring ways the work that takes place here at MIT. We have a number of different kinds of programs that are contained within the Within Sight lecture series. Tonight is about art. It's an important event that we do once a year as our gallery is renewed, and we'll get to hear from contributors and winners of the competition that ran this year as we move forward. But we have two more events this year I'd like to have you think about attending. One is in June, very much back to the front page of Science, if you will. We'll be talking about the role that the immune system plays in cancer. And then in November, we'll have an event where we're talking about the enablement of personalized medicine, something I suspect many of you have heard about, how technology is rolling forward and how that might be imagined to personalize the type of care cancer patients receive in the future. It's important to recognize the people who helped us select the images that are on display. Our judges have looked through lots and lots of submissions to select those that are on display tonight, and we're extraordinarily grateful for the commitment that they've given us now 2 years in a row to help pick the winners. And we're also wanting to recognize all those other laboratories that took the effort to submit images. There are lots of images, over twice as many images this year than we had last year, which is a trend I'm delighted to report, and I hope we'll have 3 times as many images maybe by last year. And all of them, as of today, are online available for you to view in our image archive. If you haven't visited the Koch Institute's online offering, you might find it very interesting. There are lots of images that are on display and sortable in a number of different modes. I'd encourage you to take a look there as well. Of the 10 images that are out on display in our gallery, 9 of them come from MIT-affiliated laboratories. And they'll stay there for a full year. Meanwhile, we'll open the competition again. Submissions will come in and we'll review. And next year, about this time, we'll be renewing the gallery one more time. But one of the images is here through a partnership that we have with the Wellcome Trust. The Wellcome Trust in London and the UK runs an extraordinary image competition, one of the world's most premier, I believe. And this year, we're delighted to say that in addition to us having one of their images on display, and you'll hear about that in a moment, 2 of our images are going to be in London on display in their gallery as well throughout the next year. So we're quite delighted by that partnership. Each of these images is more than just a piece of art. It tells a very specific scientific story. And they're selected based on the fact that they're not only just beautiful, but they're quite informative. Each of those panels outside has a QR code. If you happen to have one of the gadgets that can read one of those things, you can shoot a photograph of that, and it will take you to a video where the scientists/artists describe in great detail the type of work that they're doing, the type of image that they created, or the technology that was behind how they collected that image. And you might find that fascinating. If you don't have one of those gadgets, and I suspect a significant fraction of you don't have a QR code reader, you can easily find all those videos again on the offering that's online or in the gallery portal. You can put a gallery portal on your phone and be able to walk through the gallery and get all the content that's there electronically. Before going past it, I want to recognize Alex Florentino. He is our public outreach gallery coordinator. If it wasn't for Alex, the gallery wouldn't exist. He puts his heart and soul into this, and you'll be hearing from Alex in just a moment. He's the individual that also gives what I'll call the priceless Koch Institute tour. So if you want to be interested in seeing the behind the scenes of the Koch Institute, Alex is your man. And before turning it over to Tyler, I'll have Tyler tell us a little bit about what he thinks the world has looked like. Having now been here for 1 year, our director, I want to get practical just for a moment. So this within society that we have, this benefactor group, is a key tool that we use to raise the money that's required to renew the gallery. If you are interested, what you need to do is that inside your material that you were handed when you came in, there's a little tab of paper that has your name on it. You can tear that off and leave that at the reception desk, and they'll send you information about the possibility of you being interested in joining this benefactor group. And again, if you do choose to join, you have an opportunity to perhaps gain access to those images that are printed in very high quality, limited edition box sets. So I'd like to turn it over to Tyler Jacks and have him reflect with you for a moment, also welcome you in his own words, what it's been like to be in this very special building now for exactly 1 year. This is our birthday week at the Koch Institute. We have a number of things that we're doing to celebrate that, and only he can share them with you in as forceful a way as possible. Tyler Jacks.
Thank you, Robert, and welcome everyone. I see many familiar faces, but I know that there are a few of you who haven't been to one of our evening programs before. This is actually a wonderful venue to bring the Koch Institute to friends and supporters and to the general public to tell you a little bit about what we do and to give you a bit of an inside view of the Koch Institute and about cancer research at MIT, and we think interesting new developments in cancer research more generally. And I want to thank Robert in particular for all the work that he does in support of the Koch Institute, but especially in support of this program. I think Robert really deserves credit for conceptualizing this program in general and does a lot of the legwork in pulling it together. So thank you for that. This is an exciting week for us. It is our birthday week. I think it was just a year ago today, in fact, that we had the inaugural ceremony out in the courtyard. So this is our 1-year birthday. And as such, I think it deserves a song. Happy birthday to you. Happy birthday to you. Happy birthday, dear Cassie. Happy birthday to you. That was lovely. And we have 10 beautiful new birthday gifts to celebrate our first birthday. And those are the images that you have on display in the gallery this evening. And you'll hear all about those images from the scientists / artists who created them. The gallery is about really celebrating science and art, science as art, and really exploring the creative expression that underlies both of these noble pursuits. And I think the images out there are beautiful, and I think they are inspiring. We developed the gallery to stop people in their tracks as they walked past, to encourage them to come inside the doors and to learn more about what was behind those images, and in that way educate them and excite them and inspire them. And some number of them will go on to get into this field and make great discoveries of their own. So thanks to the folks who were behind the images in this round and in the past round and in the future rounds as well. We're really excited about this program. And we think that it does add a lot to the Koch Institute and gives something back to the community and represents a face of MIT that makes it a bit more of a welcoming place than you might have otherwise considered. I want to emphasize something that Robert mentioned. This program requires funding, and we've received significant funding in the past, and we're always looking for more funding. That's a theme that runs throughout all of my remarks. We're always looking for more funding. But it's true of the gallery as well. So we have a benefactor society, the Within Society, and to encourage you to become a supporter, we offer those books of limited edition prints of the images that appear in the gallery, and they really are wonderful. We'll throw in some Ginsu knives as well. But I do encourage you to take that opportunity seriously. So we've been here for a year, and it's been a very exciting year. As many of you know, the Koch Institute is special in the sense that it brings together scientists, biologists, cancer researchers, and engineers who themselves have a cancer orientation under one roof, drawn from across the MIT campus, to pool their talents and to work together to discover new information about cancer and to develop new approaches to control the disease. And I'm happy to say that what is really still an experiment, would it make sense? Would it be productive to bring these diverse groups of people together in one building? The experiment is paying off in spades. Paula Hammond, one of our engineers, is nodding enthusiastically. I like to see that. We have, in fact, begun to develop a new community here in the Koch Institute, where chemical engineers are talking to molecular geneticists, and biological engineers are talking to cell biologists, and they're creating a new language, a common language of discovery. They are indeed pooling their talents and taking us in new directions that we simply could not have anticipated before we moved in. And so this is a tremendously exciting opportunity for us, and we're extremely grateful to the many people who have made it possible. Just to walk in the door and to go up the elevator and to hear the conversations bubbling up around us is incredibly exciting. You know, we were inspired in the early days from discussions that we had with Susan Hockfield shortly after her arrival, as she began to note the opportunities for this convergence, as she talked about, this convergence of life science and engineering, these two fields coming together, the opportunity of working on common problems collectively. But what we have here in the Koch Institute goes beyond convergence. What we have here now is a confluence. These great streams of engineering and science are now flowing together, and new ideas are literally bubbling up all around us, inspired by the conversations that take place and random interactions in the hallway and in the elevators and in the bathrooms. It's remarkable to see this generation of innovation that's taking place by virtue of bringing these folks with different perspectives together around a common problem. So we're extremely excited about what's happening here and extremely excited about what can happen in the future. Just to mention a few examples, and there are many, many that I could choose from, just a few examples of progress that has happened just over the last year. Our colleague Angelica Ammon, who studies the cell cycle, the processes by which cells make decisions to divide or not divide, and tries to understand the contributions of aneuploidy, the inappropriate amount of DNA, of chromosomes that cancer cells have to that process. She's undertaken a genetics, small molecule screen to find drugs that can specifically kill cells that have too much DNA. Now that's relevant because cancer cells almost always have too much DNA. Your normal cells have the right amount, and almost invariably cancer cells have too much. So if we could find drugs that selectively kill cells with too much DNA, that could represent a very important new direction in cancer treatment. And she has found such drugs, and those concepts are now being developed for preclinical and soon clinical testing. Metastasis. Metastasis is a major killer. 90% of all cancer deaths are due to the metastatic phase of the disease. And yet, it's a phase that we don't understand very well. Many investigators within the Koch Institute have made major discoveries over the last year about the biological processes that go wrong as cancer cells escape from the primary tumor and move throughout the body. And these are identifying new targets to inhibit, to prevent the spread of metastatic cells or to kill those metastatic cells in their foreign site. Our engineering colleagues have developed new imaging agents that will allow us to find cancer cells that have made it to those distant sites in very small numbers and to diagnose cancers at earlier stages when surgical interventions might be more successful. And our nanotechnology colleagues have developed new methods for delivering cancer drugs and other materials directly to cancer cells. And in the story that I like to tell these days, a concept that was born not very long ago, just in about 2003, in the laboratory of Bob Langer, and funded in that first phase with institutional funds that come from supporters like you. That project got initiated. Federal funds were brought in as it began to be more successful and had to be expanded. It was spun out into a company called Bind Biosciences, and the first clinical trials of this nanotechnology-based cancer therapy were launched last year. Therapy has proven to be very safe, highly effective in terms of delivering doses of cancer drugs, and in very preliminary studies, some patients are showing clinical responses, from concept to clinical testing in just 7 or 8 years. Really quite remarkable. It shows the power of bringing cancer science and new technologies together. And so there are many stories of our success. Robert's computer has gone to sleep. Hopefully you haven't as well.
We'll turn back on.
Many stories of our success, and we will be hearing about many, many more in the year ahead and in the years ahead. And so thank you for coming and sharing tonight with us. I want to close by making the point that the Koch Institute grows out of a great tradition in cancer research here at MIT, and specifically out of the old Center for Cancer Research that existed at 40 Ames Street, our old home, the old Cancer Center, and to commemorate the wonderful contributions that took place in the Cancer Center, we've created a memorial plaque, partly to Salvador Luria, who was the founding director of the Center for Cancer Research, but also to all the faculty who worked there and all of the important work that they did in establishing cancer research on this campus and really creating the foundation for what is modern cancer research today. I encourage you to take a look at the plaque, and there's one of those QR codes there, and you can use that to go to a terrific video that our friends at Feinstein Keene put together that really does capture the essence of the Cancer Center. For those of you who don't know, if you have a cell phone, you can actually put a QR code reader on it. It's very easy to do. If you have the MIT app, for example, you can do it directly. You do have to know how to use your camera, and I was trying to do it earlier today. And one of my colleagues pointed out that I actually have to point the lens of the camera at the QR code, which is very important. So I'll stop. Thank you again for coming. Tonight is about science and about art. And so for that, I'm going to turn things over to Alex Fiorentino, who, as you heard, is responsible for organizing much of what you see outside. Thanks very much.
Thank you, Tyler and Robert. Both of your comments were great. Okay, so I'm just going to give a quick introduction to the format we're going to be using for the rest of the night. It's a little bit unusual, so kind of buckle up and be ready. Basically, what I've asked our great scientists, artists, engineers to do is take months or years of their research and boil it down into 2 minutes for all of you, and also make it into 2 minutes that anyone can understand. So not a small task that I've given them, but I think they'll do admirably from what I've seen of them already over the last few months. And so inevitably, from a 2-minute talk about the kind of science and engineering that these folks are doing, you'll have some questions. If you do, hang on to those till the end. At the very end of the sequence of talks, we'll bring all the speakers back up, and you'll have a chance to ask any questions you'd like of them. So without further ado, I will kick things off with our first scientist artist, and that is Benjamin L. Larson from the Langer Lab right here on the 6th floor of the Koch Institute. And Benjamin is going to talk about his image right here, which is entitled Building a New Hip, Cartilage Grown on a Woven Polymer.
Thanks, Alex. 12 years, 2 minutes, here we go. So we were asked to come up here and tell you a little bit about our picture and about why we took the picture, why it's important to us. And to me, what's so important and what's so cool about it when I took it, why it was just like something rose up inside of me that this is going to be really important, this is really amazing, is that when I take a section, a small slice of human cartilage, a hip or a knee, cadaveric tissue, people aren't alive anymore. When I take that tissue and stain it with this franano that I stain it with, this is what I see. Only this is not a cadaveric tissue, this is not from a human, this is stuff that we grew. And we grew it out of stem cells, we grew it out of polymer, and it's just amazing. On the other side is bone. And we have a beautiful transition zone in between. Really cool stuff that we're doing here at the Koch Institute on the 6th floor. Why we wanted to do this was because many cancers are non-metastatic and they can be removed, and that's great. But people need a restorative function, and so the amputees, the osteoarthritis, the trauma patients, you need restorative function to hip or knee that's been removed. And so we're hoping to replace that with cartilage and bone grown in the laboratory here at MIT and in the Koch Institute, thanks to donors like you. And so this here is just cartilage that we grew. This is focused more on the cartilage side. Our polymer, you can barely see it up in the top left. The interwoven polymers are since then biodegraded, and so you can't see them anymore. The cells are the green, and the cartilage is the red. And again, it's just to me and to other more osteocyte surgeons, it just looks beautiful. It is cartilage. It is what you'd see in the body, and that's exactly what we want to see. The only difference is it's grown from scratch, and it's grown here in our laboratories to hopefully help restore function to people in the near future.
So up next, we have Pranit Namburi, who is a graduate student in the Department of Brain and Cognitive Sciences here at MIT, and his image is neural mapping, understanding how the cerebellum is wired.
Hi, good evening. I'm Pranit, and I'm going to tell you a little bit about this image. So the human brain has about 100 billion neurons. So what you see in this image are two types of brain cells. One's highlighted in red and one's highlighted in green. So these neurons have the ability to process and relay electrical signals. Understanding how these neurons connect with each other is one of the most important ingredients towards understanding how the brain works and towards understanding human behavior and cognition. So this image was taken when we were doing an experiment to understand this wiring in a part of the brain known as the cerebellum. When I see such images, I'm amazed as to how evolution can give rise to something so beautiful, complex, and organized and functional like the brain. Human curiosity and relentless pursuit to understand ourselves and the world around us has given us technology which allows us a peek into the brain. Relevant to this image, this technology includes genetic engineering, which allows us to take attributes from jellyfish and algae and put them into parts of a mouse brain. It also includes technology that allows us to focus laser beams and light into brain tissue. So hopefully, such relentless pursuit will one day give deep insights into how the brain works and into human nature. Thank you.
So up next we have Salil Desai, who is from the Bhatia Lab here in the building as well, and his image is motor control. How does a cancer cell power its movement? And I encourage you, Salil, I think, to hold the mic next to us.
You have the laser pointer, right?
Sure, yeah, it's just tucked right behind you.
Thanks, Alex. So the goal for this work was to study the spread of cancer at the microscopic level by being able to observe individual cancer cells and understand how they move. So what this picture shows is 3 individual breast cancer cells, if I can get this laser pointer to work, shown here, that are moving down beginning to move down these microscopic channels. And just to give you a sense of the scale of these channels, each of them is about 1/10 of a width of a human hair, 10 microns. And what we're trying to do is place these cells into these different channels and observe how their components organize as the cell moves. And specifically what I was interested in is looking at mitochondria, which are shown here in red, which are the energy centers for the cell. So I'd like to know how these energy centers are localizing and powering the cell forward. So if I were to use an analogy of these cells being cars that are zipping down lanes in a freeway, then I'd like to know, well, how many of these cars or cells are front-wheel drive? How many are rear-wheel drive? Are any of them, all-wheel drive cells? And more importantly, you can start asking some interesting questions like, are the front-wheel drive cells more persistent? Do they invade different tissues? Can they move further in the body? And we can really start beginning to answer these questions with this type of technology. And what I like personally about this image besides the colors, is that it sort of evokes a sense of being new possibilities. So we can do this with one type of cancer cell. These are just breast cancer cells. We could do this with pretty much any other type of cancer cell, get them to move in these channels. And the more we understand about the underlying mechanisms of how the cancer cells move, the more we can think about how to stop them or put the brakes on them so that we can start fighting the spread of cancer. Thanks.
Thanks, Liam. So this next image actually is one that Robert referred to that comes from the Wellcome Trust, and not come from here at MIT. And so the creators of this image can't be here with us today, but we'll just take a moment to look at their image. They are Ferdinand Federici, Tim Rudge, PJ Steiner, and Jim Hasteloff, all from Cambridge University in the UK. And this image shows what we call bacterial conversations. So this is self-organizing communities of Bacillus subtilis. So if you look closely, it's maybe easier to tell out there when you get a little closer. You can see that this is made up of many, very small, they look like grains of rice here, but they are bacterial cells, and they're organizing into these patterns because of the way they communicate with each other, sort of self-assemble. Okay, so we'll move on to the next speaker now, and that is Rakesh Karmacharya. And Rakesh joins us from the Koch Institute as well as Mass General and McLean Hospital. And his image is entitled Modeling the Mind, Neurons Derived from Skin Cells.
So what we are trying to do is study the neurobiology of severe neuropsychiatric illnesses such as schizophrenia and bipolar disorder. A fundamental problem for us is the inability to study the tissue of interest to us. Unlike if you're studying liver pathology, where you can get a biopsy, we understand that we cannot get brain biopsies from the patients that we're trying to study. So we've been limited in studying postmortem brain specimens or doing imaging, which has its advantages, but we've been severely limited in that aspect. So we were fortunate to have this advanced stem cell technology in the last couple of years that allows us to go from any cell in the body, in this case a skin cell, and reprogram it to becoming the equivalent of an embryonic stem cell. So what we do is take a skin cell from the patient and then make what are called induced pluripotent stem cells. These are for all practical purposes like embryonic stem cells. The advantage now is that once you have those cells, you can make any tissue in the body. They have the potential to make any tissue in the body that is of interest to us. And in our case, it's neuronal cells. So first we take those induced pluripotent stem cells, make them into neural stem cells, and then let them differentiate into the different kinds of cells that comprise our brain. So in our picture here, this was something that was differentiated over 2 months. We let them develop over 2 months from the neural stem cells. And the green ones show the neurons. And then the ones in the background that you can see in red are what are called the glial cells, which are often thought of as the support cells, but they also have a pretty important role in the way our brains function. The blue one just shows the center of all the cells. That's where the nuclei are. And it's amazing how our brains are three-dimensional structures, but even in a two-dimensional plate in the dish in the lab, we can see the beauty of all the connections that the different neurons are making between themselves. And the idea is we're trying to see when we make those images from patient cells as well as healthy controls, are there differences that we can observe in these living, growing neuronal cells? In the event that we can find robust differences between those control ones and the diseased ones, then the idea would be to try and do screens with small molecules to try and see how we can correct those deficits, correct those differences that we can see in the diseased cells that we don't see in the healthy cells.
Thank you. Up next, we have another image. Unfortunately, Brett couldn't be here tonight, but I understand that his advisor, Dr. Edelman, is going to come say a few words about Brett's image, which is it was created in Dr. Edelman's lab, and is entitled "Crossing Over: Cells Move Through an Epithelial Bridge."
So I have the privilege of representing a student, which doesn't often happen. Brett's a brilliant student, he's a brilliant postdoc, and he really very much wanted to be here tonight. But it's also the opportunity to thank the leadership of the Koch Institute for their pioneering work in really bringing the MIT campus onto the forefront of the integration of science and engineering. Our laboratory uses engineering principles to study how fundamental issues in cell biology affect the development of organs and then explain basic disease. We asked a very simple question. If I asked all of you sitting on this side of the room to move to the other side of the room, would we still have the same interaction? All cells, all organs, start with the same fundamental building blocks, and the wonder is that we create these beautiful people that we are. And so what Brett did is he asked a very simple question. If I just take two cell types and I put them together, what happens? And they found that like communities, cells tend to isolate themselves, but they find magnificent ways of bringing themselves together. And what he discovered using fairly sophisticated imaging technology is that a cell can create its own bridge. And you see these cells here, highlighted in green, are epithelial cells. And they're hurtling over endothelial cells, a different kind of cell. This process is very orderly, very arrayed in normal tissue, and it grows and it goes entirely disarrayed when there's disease like cancer. It's a spectacular image. It's an image which took extraordinary feat of engineering to capture, and it has profound ramifications for the way we think about normal and disordered development. And it's really an honor to represent a student in an assembly like this. Thank you.
Thank you, Dr. Edelman. So the next talk will be from Peter Demuth, who is from the Hammond Lab and Irvine Lab here at the Koch Institute. And Peter's image is entitled Immunophores, Microneedles for Delivering Vaccines into the Skin.
Good evening, everyone. I just want to describe this image to you in a simple way. So basically what you're looking at here is what we call a microneedle array. And it's essentially exactly what it sounds like. It's an array of needles that are less than 1 millimeter in height. And this image was taken as part of an effort by our lab to create a non-invasive vaccination strategy that would overcome a lot of the challenges that are inherent to the use of hypodermic needles. And so the idea here is that this microneedle array could be simply and easily placed on the surface of the skin to deliver a vaccine in a way that is safe, inherently more effective as a vaccine, easy, convenient, more cost-effective, and my favorite, inherently painless. So why is this image beautiful? To me, it represents our ability as materials engineers to create strategies on a very small size scale that overcome some of the problems that we face in our world today. And coupled with that, I think it's beautiful because it represents a platform that has the potential to really change the way we think about vaccination, has the potential to address challenges that current vaccines can't really address. One of the most promising, I think, is the ability to deliver vaccines to areas where they're perhaps most needed, to areas of the world where the burden of disease is the most heavy. But currently, the technology that we have make it difficult to get those vaccines to those areas of the world. So thank you.
Thank you, Peter. So the next speaker will be Laura Adolfi, who is also from Dr. Edelman's lab here in the Harvard MIT Division of Health Sciences and Technology. And Laura's image is entitled Form and Function, Using a Curved Surface to Change Cell Behavior.
Good evening, everyone. So what you are looking at in my picture are endothelial cells, like the cells lining the blood vessel, that are stained in red for their skeleton, for their structure that makes them interact with the environment outside their body. And what is really important for them is that endothelial cells and therefore the blood vessel are present in any tissue in our body, and therefore understanding their their function and understanding how we can modulate their outcome can be extremely helpful in helping us fighting many diseases, including cancer. So the aim of our project was to figure it out is if there is a way to change their behavior other than the classically used using some drugs. So the hypothesis was that in the glial cells inside the body, they are experiencing an environment that's not flat, but is more complicated. And so if we can recreate that environment in vitro, we can induce some cell line to act more healthier. So that is what we did, and the way that I think this picture is really powerful is because it was the first time that we can actually see from a microscope that our hypothesis that the structure of this environment is important was real. And that is because the matrix where we see the cells has some dimension that is subtly smaller than the cells itself. So the cells cannot be flat anymore, as many studies have been using, but they can achieve themselves a three-dimensional morphology. And that was just the beginning of the project, and nowadays, that is, after 3 years of my postdoc, we have more evidence that that is indeed the case. We can treat disease where inflammation is an issue just by seeding healthier endothelial cells in a more controlled environment. And then they can act and reduce the inflammation. So what makes this picture more important is that as a personal side, that was the first picture of my project. And when I moved here, I was just a classically trained engineer. I worked with material, and that was the first time that I combined material science with biology and being able not to kill the cells and be able to see them. And it was quite an achievement. But also, we just don't only use the cells in the lab, in Petri dish, but we also can make them available to patients and kind to help them have a better life.
Thank you, Laura. So this second to last image is from another speaker who wanted to be here, but unfortunately couldn't make it. And that's Miji, who is a researcher in the Van Hoevenarden lab in both the Department of Biology and the Department of Physics here at MIT. And Miji's image is this, which is entitled Breaking Rank, How Identical Cells Take on Different Fates. And I always, when I see this image now, I always think of how Miji described it to me in the interview we conducted for the website, where she said this is a humble worm that she spent months and months working with. And it's this fairly simple organism, not very many cells, but that tells biologists, including me, a great deal about how life works. And in this case, she was looking at, you can see this cell right here is in the process of moving from its place in kind of this sequence back further toward the tail of the worm. She's trying to understand exactly how the signals work that tell this cell to move backwards and another one on the other cell to move forwards when initially they seem to be identical cells. So that's Miji's image, and then that will move on to the last speaker of the night, which is Christopher Turner, who is from the Heinz Lab here at the Koch Institute. And Chris's image is building a biohighway. How does a lymphatic cell develop?
Good evening. So my image actually shows a lymphatic vessel. So this big green structure here is a lymphatic. And in red you can actually see red blood cells that are flowing through blood vessels in the skin. Now, for those of you who have not heard of lymphatics, lymphatic vessels actually absorb fluid from tissue and actually pass it back into the blood supply. They're also important for the movement of immune cells. So immune cells actually travel through this pipeline of lymphatic vessels around the body. We're particularly interested in them because it's been actually known for hundreds of years that tumor cells can also use these lymphatic vessels as a pipeline to spread around the body. So by learning more about these vessels and potentially by stopping primary tumors from recruiting these pipelines, we might be able to stop primary tumors using this highway to spread around the body. This image actually, I get quite fond memories when I look at this picture because I actually took it around midnight. It's actually a Friday night when I should be thinking about the weekend. My wife was calling me out, telling me to come home. I was in a dark microscopy room. But it actually took me about 6 weeks to get to the stage of this kind of image. So it was a lot of work, and finally I cracked it. I got my first real look at a lymphatic vessel. And it's only due to advances in microscopy that we can actually image deep into tissue now and actually visualize blood vessels and lymphatics in vivo. So, thank you.
So if all the winners would come up and gather up here, we have some time for questions. And actually, how about a round of applause for all of them? And if you have a question, please raise your hand and we'll bring the microphone to you. Any questions?
So who picks the colors?
Oh.
Yeah.
Who picks the colors? Yeah, so actually most of these images I think we can say were the same techniques we use antibodies. So antibodies can recognize almost any protein in the body. And then we usually attach some kind of fluorescent protein to that. So that's how we can get these kind of images. The actual color, we can choose what we like. So if you have a preference for next year, we can make it.
Any others? I have one question. Peter, I know the answer, but I love the answer. So how do you get them so sharp? Those needles.
Yeah, so the process that goes into making what you're seeing in the image is fairly simple, actually. It involves a molding technique that you would typically think about for modern manufacturing. The process involves taking a piece of rubber and using a laser to very carefully cut cavities into it to form the mold. We then use polymers that we melt at high temperature, and these flow into the mold. And upon cooling, we're able to remove those from the mold, and they take on the shape of the mold itself. So the sharpness comes from the machining that we perform with the laser.
Well, why are they all perfectly identical to each other?
So that is not actually an easy thing to do. It's actually quite difficult, and you have, for this image, you probably have hundreds of images that look not nearly as good as this. So it's a process that takes a lot of optimization and a lot of fine control over the laser itself. And that just goes back to, again, why I think this image is so arresting, that we can have this amount of very fine control. I think it's just phenomenal.
Any last questions? No? Well, we should give them one last round of applause, I think. Our evening ends with a little bit more networking. So all of the science artists will be available to chat with you specifically about what they do. There's refreshments out for another 30, 45 minutes. Thank you for coming. I hope you come again. We have another minute or two. Thank you.